Hello everyone, my name is Hatim Abulola and I'm a graduate student in the biochemistry program in Dr. Gage lab, and I do real-time imaging of calcium transients in skeletal muscle fibers to study the spatiotemporal dynamics. So calcium plays a major role in muscle contraction. When the signal comes from our brain to contract the muscle, it comes in the form of action potential, which will release calcium from the intracellular stores through the calcium channels to the cell cytoplasm, which will lead to muscle contraction. And I'm interested in studying the spatiotemporal dynamics of calcium transients as it moves through the cytoplasm. To visualize calcium in muscles, we use fluo4AM, which is a calcium indicator masked with ester groups. The ester groups will allow the entrance of fluo4AM to live cells. Then cell esterases will break the ester groups, making fluo4 free to bind to intercellular calcium. Here I'm showing a muscle fiber that is electrically stimulated using a 15-volt, 25 millisecond pulse duration at a frequency of 1 pulse per second. And as you can see, there's a calcium pulse associated with each stimulation. Then I did some experiments to confirm that the observed calcium is coming from the intercellular stores. So first I treated the extracellular medium with EGTA, which is a chemical that takes away calcium from the extracellular medium, and I still see calcium fluctuation on the fibers. Then I used calcium channel blocker to block the calcium release from the intracellular stores, and as you can see, the calcium response is eliminated after blocking the calcium channel, and this tells me that the calcium I'm seeing is coming from the intracellular calcium stores. Here I'm showing a 2D intensity plot of calcium transients concentrations as the muscle gets stimulated with electrical stimulation. The chart in the top left corner shows the intensity of calcium from high red to low blue. Then I calculated the velocity of calcium transients using the distance between the region of highest calcium concentration to the edge of the calcium wave. Here we show three successive pulses, and as you can see, the average velocity decreases with successive pulses. Each velocity was calculated using the three separate distances from the center along the front of the wave. In conclusion, calcium fluctuations can be observed in electrically stimulated skeletal muscle fibers, and fibers responded to stimulation even after EGTA treatment, which removed calcium from the extracellular medium. Also, fibers do not respond to calcium after calcium channel blocker treatment. Finally, the velocity of calcium transients are decreasing with repeated stimulation. Finally, I would like to thank my advisor, Dr. Gage, and my colleagues in Gage Lab and the UMOV Center for the support and help in my research.